Welcome back to Chapter 18. Today we're covering Section 18.5 on vaccines. This is the final component of adaptive immunity. By the end of this video, you should be able to 1. Compare the different kinds of artificial immunity, 2. Explain the difference between variolation and vaccination, and 3. Describe major vaccine types, including their advantages and disadvantages. This picture is actually from the beginning of Chapter 18, but it's a picture from 1953 that shows a polio ward filled with iron lungs. So before we had widespread vaccination, thousands of patients would end up paralyzed and unable to breathe independently. So this slide really sets the stage for understanding why vaccines have dramatically changed medicine. Vaccination creates artificial active immunity. It stimulates a primary immune response, so the body produces memory B and T cells. This allows a very strong secondary response if you're exposed to that same pathogen in the future, and this prevents us from having to experience and suffer through the disease from a natural infection. Adaptive immunity categories break down into active versus passive and natural versus artificial. In active immunity, the body is going to produce its own antibodies against the pathogen. Whereas in passive immunity, antibodies are being transferred from another source, another human or animal that was previously exposed to the pathogen and made antibodies that are being transferred to the patient. If we put these combinations or categories together, we get natural active immunity. This is when you get sick because you're exposed to that pathogen and then you recover. Natural passive immunity is when IgG antibodies cross through the placenta from the mom to the fetus, or IgA is given to the infant through breast milk. For artificial passive immunity, this is through injection of antibodies, and artificial active immunity is through the traditional vaccination route. So let's go through those 4 categories more closely. In natural active immunity, this is immunity acquired through actual infection. So the immune system will fully activate, generate those memory B and T cells for future secondary response, and this provides long-term protection. Examples include recovering from influenza, COVID-19, or chickenpox. In natural passive immunity, infants receive passive protection. IgG can cross the placenta and protect the fetus, but also can protect the infant up to around 6 months after birth. Infants can also receive IgA through breast milk, which protects mucosal surfaces. But this is temporary protection. When you're receiving antibodies, there are no memory cells being formed for future responses. In artificial passive immunity, we're giving pre-formed antibodies from humans or animals that were exposed to the pathogens and made antibodies to those pathogens. So imagine you're receiving those pre-formed antibodies, and this is useful for post-exposure treatment for situations like rabies, hepatitis A or B, or varicella, but it can also be used for toxin-mediated disease like tetanus or botulism or diphtheria. And these antibodies provide immediate protection, but again, it's short-lived because no memory cells will be formed. And finally, we have artificial active immunity. This is the goal of vaccination. We're giving antigens in a safe form so the immune system develops memory. And this graphic summarizes those natural versus artificial and active versus passive categories. So passive, we can see we're receiving antibodies either naturally or artificially from another animal or human. In active immunity, we're either getting sick from the pathogen directly or getting immunity or acquiring immunity through a vaccine. This first checkpoint asks, What's the difference between active and passive immunity? So in active immunity, the host's own immune system produces antibodies and memory cells. So this gives us long-lasting protection, and it can occur through either infection directly from the pathogen or through vaccination. For passive immunity, the host is receiving antibodies made from or made by someone else, another person or another animal. This provides immediate protection, but it's short-lived because there's no memory cell formation. So examples include getting maternal IgG through the placenta or breast milk IgA. For the second question, what kind of immunity is conferred by a vaccine? This is artificial active immunity. So a vaccine will stimulate the host's immune system to produce antibodies and memory cells without causing disease. Here we can see a graphic to summarize the two. Passive immunity spikes quickly, so it gives you immediate protection, but it fades fairly quickly as well. Whereas active immunity builds up more slowly, but lasts much longer and can be boosted through these boosters. Herd immunity occurs when enough individuals are immune that a pathogen cannot spread. So if we look at this example, there's no herd immunity. Everyone or most people are susceptible. It's easy for the disease to spread. But if most individuals are immune, as shown in green, it is very difficult or impossible for the disease to spread. Most diseases, and this really varies, but most diseases require an 80 to 95% vaccination rate for that population for herd immunity to be possible. If the disease is highly contagious, like measles, we require at least 95% or greater vaccination rates. And herd immunity is important for patients like SCID patients, which we'll talk about in the next video, who cannot be vaccinated. So populations that follow the recommended vaccination schedule provided by, for instance, the American Academy of Pediatrics or the CDC, really provides our group, our society, with herd immunity. And that makes it really hard for a pathogen to spread when most people are resistant due to the vaccine. And this always kind of reminds me of zombie movies. If you've ever seen Train to Busan or similar movies, these are almost always caused by some kind of virus. And imagine if there was a vaccine for the virus, right? For that zombie virus. Then the movie would be really boring because very few people would become zombies overall. And just for fun, the CDC has this zombie preparedness graphic novel. If you're interested, you can download it. This is the website over here, and it's really talking about emergency preparedness. But they do have within the story CDC scientists that develop a vaccine. Imagine if everyone or most individuals were vaccinated, that would provide that herd immunity to prevent spread. Our book also has a section on vaccination hesitancy, and vaccine hesitancy often arises from misconceptions, ethical concerns, or misinformation. The problem with vaccine hesitancy is that lower vaccination rates often correlate with a resurgence of preventable diseases like measles and whooping cough, and unfortunately generally leads to an increase in death rates. States with very strict vaccination policies, like West Virginia, Mississippi, or California, have the lowest measles transmission, for instance. So this really raises important public health and ethical debates. One of the biggest probably contributors to the vaccine misconceptions was probably due to Andrew Wakefield. He was a physician who published a paper in this famous journal, The Lancet, in 1998 that falsified data and claimed that the MMR vaccine caused autism. Unfortunately, at the time, it was really amplified by different media sources, as well as this anti-vaccine group. This is probably one of the largest anti-vaccine groups here, the National Vaccine Information Center. But once the Lancet investigated Andrew Wakefield and this paper more carefully, they found out that there was fraud, serious misconduct, and fake data. So Wakefield eventually lost his medical license, and since then, hundreds of high-quality research studies have found no link between vaccines and autism. However, there's still residual misconceptions out there because of Andrew Wakefield. So this stays or remains a major source of misinformation. And if we look closely at vaccines, it's really interesting because although the number of vaccines recommended has increased, the total antigen exposure has actually decreased. In the 1900s, children were only given one vaccine, which was against smallpox, by the time they were 2 years old. In the 80s, there were more vaccines like tetanus, measles, mumps, rubella, etc., recommended by the time kids were 2 years old. Today, there are at least or around 14 vaccine types that are given according to the recommended schedule by the time children are 2 years old. But the total antigen counts are fewer. So in the 1900s, that single smallpox vaccine had around 200 antigens. But today, our entire vaccine schedule amounts to only around 130 to 150 distinct antigens in total. And today, modern vaccines are much more targeted and safer. Variolation is no longer practiced, but back then it was an early method to prevent smallpox. So what they did was they took smallpox patients and took some of their scabs or pustule material and infected or introduced this to other people to protect them. Sometimes this offered immunity through mild infection, but in other times it caused severe or fatal infections. The practice also could spread smallpox. And this really set the stage to search for safer alternatives. And the reason this was called variolation is because the virus that causes smallpox is the variola virus. Edward Jenner discovered that cowpox, a milder disease, protected against smallpox. So here we have a painting of Edward Jenner, and there is a cow and a milkmaid in the background. On the right, there are lesions on a patient that is infected with cowpox. This is a zoonotic disease caused by a virus that's very closely related to the one that causes smallpox. So because cowpox antigens are similar to variola virus, which causes smallpox, this offered cross protection. And this drastically reduced the risk, like we saw in variolation, and led to the modern concept of vaccination. And vaccination comes from that Latin word for cow. This checkpoint asks, what's the difference between variolation and vaccination for smallpox? So variolation was deliberate exposure to infectious smallpox material through those scabs or pustules, and it could cause a mild smallpox infection that led to immunity, but it was also very risky because it could also cause that full-blown smallpox, could cause death, or transmit smallpox to others. For vaccination, this was discovered by Edward Jenner. He used the cowpox virus, which is antigenically similar but much milder. So this produced immunity without causing smallpox. It was much safer and did not risk the spread of smallpox. So this is kind of related to the second question, why vaccination is less risky than variolation. Again, because vaccination uses non-smallpox material, right? It does not cause severe disease. It causes cowpox. And in vaccination, we're eliminating the risk of triggering that dangerous smallpox infection. We don't have to worry about transmitting smallpox to other people. Overall, we get effective immunity with lower danger. All right, now we're going to switch gears and look at the different types of vaccines that are available. There are 5 classical categories of vaccines and a few new categories as well, and we'll go through each one. Live attenuated vaccines contain weakened pathogen. So the pathogen is still infectious, but weaker. So these mimic natural infection and produce strong, long-lasting immunity. It activates both the cellular and humoral immunity divisions. But there are some risks because you're still using live pathogen. So the risks include mild symptoms, reversion of the attenuated version to the full-blown strength pathogen, and it's sometimes not safe for immunocompromised or pregnant patients. Examples include the MMR vaccine, varicella, yellow fever, or the nasal spray flu vaccine. Inactivated vaccines are also called killed vaccines, and the pathogen is killed. It cannot replicate. This is safer, but produces weaker immunity, and you often need boosters. Examples include hepatitis A vaccine, the inactivated flu vaccine, which is usually the typical flu shot. Vaccines against cholera or rabies are examples as well. Subunit vaccines only use purified antigens, so just a part of the pathogen. For example, here is the live attenuated virus, and for the subunit vaccines, you're only taking some components of this virus. It's very safe with fewer reactions, but immunity overall is generally weaker and may need boosters. Examples of subunit vaccines include the hepatitis B vaccine, HPV vaccine, that's a human papillomavirus, or acellular pertussis, and pneumococcal pneumonia vaccines. Toxoid vaccines target bacterial toxins, not the bacteria itself. And toxoids are inactivated bacterial toxins. So these will induce the production of antibodies in our bodies that will neutralize toxins if you ever encounter them in the future. But they don't prevent infection. So they're not going to prevent infection by the bacterium in the future, but it will prevent the effect of the toxins, since you'll have produced antibodies or memory cells that can produce antibodies against the toxin directly. And examples of toxoid vaccines include vaccines against diphtheria, tetanus, or pertussis, and these are components of the DTaP vaccine. Conjugate vaccines are designed for polysaccharide encapsulated bacteria. So it turns out that infants can't mount a strong response to or against polysaccharides by themselves. But if you attach them to a protein carrier, this is called protein conjugation, it'll convert the T-independent response that usually occurs with polysaccharides to a T-dependent response. And this is now going to become a T-dependent antigen and allow memory and class switching that doesn't usually occur with T-independent activation. And examples of conjugate vaccines include vaccines against Haemophilus influenza type B, or pneumococcal conjugates, or meningococcal conjugates. This table from our book shows us those 5 major classes of vaccines, their description, advantages and disadvantages, and some examples for each. This first checkpoint question asks, what is the risk associated with a live attenuated vaccine? So live attenuated vaccines contain weakened but living pathogens, and in some cases the organism can cause mild symptoms. It might spread to immunocompromised individuals, and it's very rare, but it is possible for it to revert, like go back to the more virulent form. Sometimes live attenuated vaccines may not be recommended for severely immunocompromised individuals or pregnant patients. The second question asks, why is a conjugated vaccine necessary in some cases? So polysaccharide capsules of some bacteria are T-independent antigens. And remember from previous videos, this means that it elicits only weak IgM-only responses. There's no memory cells formed, and they're also poorly immunogenic in children under 2 years old. So the solution is using conjugate vaccines. This is when we link the polysaccharide to a protein carrier that will convert it to a T-dependent antigen, and this results in a stronger immune response. Now you can also have class switching from the B cells so they can make IgG antibodies. You'll form memory B cells, and this provides effective immunity in infants and young children. Our book also has a section on DNA vaccines, but it's a little bit outdated. So DNA vaccines deliver plasmid DNA that cells can take up and translate into antigen protein. And this will activate both cellular and humoral immunity. There are some modern improvements that go past what the book includes, including electroporation and delivering these DNA vaccines through nanoparticles. Overall, they're very safe, and we have even a COVID vaccine example that is a DNA vaccine. But usually what happens is you need electroporation. So the vaccine is injected into the skin, you're introducing the DNA, or it can be introduced into the muscle as an intramuscular injection. And immediately afterwards, you have to have this special electroporation device that you place directly onto the skin that delivers a brief controlled electrical pulse through the skin, and that will temporarily open up the membrane pores in the cells at the injection site to allow the DNA into the cells so the antigen can be expressed. Today we also have messenger RNA vaccines. So let's pretend we have this messenger RNA vaccine against COVID, and we're taking the RNA that encodes a spike protein on the virus, putting it into a lipid nanoparticle. That is going to be introduced. That RNA will be introduced into our human cells. We're going to read the messenger RNA and translate that to spike protein, and we'll make antibodies against that spike protein. So this is rapid to develop. These are faster to produce, and they're very effective. There's no genomic integration, like RNA is not going to integrate into our DNA. And some common examples are the Pfizer and Moderna COVID-19 vaccines. Another more recent type of vaccine are our viral vector vaccines. This uses a harmless viral vector, so we've taken a virus and we've changed it so that it's no longer infectious, and we're going to use that viral vector to deliver genes, antigen genes from another pathogen. This will induce strong T cell responses, and it can often be effective with just a single dose. And examples of these viral vector vaccines include the COVID-19 vaccine from AstraZeneca or Johnson & Johnson, or the Ebola vaccine. All right, now that we've talked about all the different types of vaccines, I wanted to go back to this chart and talk more about the differences between active and passive immunity. So here we have active and passive immunity. In active immunity, you're given the antigen and you're going to make your own antibodies against that antigen. Whereas in passive immunity, you're given antibodies from someone else. In each case, there are natural and artificial types of immunity. For natural, this would be infection by the pathogen that gives you natural active immunity. Artificial would be through those vaccines we just talked about. In passive immunity, natural is maternal antibodies that are received. Artificial is antibodies from someone else that are injected. The time to protection and duration is very interesting. For vaccinations, for instance, it takes time to become protected against that pathogen, but it gives you long-term protection because of those memory cells that are formed. Whereas in passive immunity, as soon as you get those antibodies, you are immediately protected, but it's short-term because those antibodies don't last that long. Maybe weeks to months, there are no memory cells formed for the future. When do you get vaccination? Usually, hopefully before you get sick, but you can also get it after, right? You get active immunity after you're infected to a pathogen naturally. And for passive immunity, it's usually after exposure. For example, after you're exposed to rabies. And then the examples we mentioned earlier are found in that last row. All vaccines have potential side effects, but overall they are overwhelmingly safe. Live vaccines give you the highest immune response, but they are also higher risk compared to the other types of vaccines. Whereas subunit or toxoid vaccines have the lowest risk, but they also give a weaker immune response and often need boosters. So there is a risk-benefit consideration, but in terms of protecting the population and protecting others, we really strongly favor vaccination. And if you're interested, these are the current vaccination schedules for children, adolescents, and adults from the AAP, American Academy of Pediatrics, and the CDC. And here's a screenshot of that immunization schedule from the American Academy of Pediatrics. Hepatitis B, for instance, is a subunit vaccine, so you can see it requires boosters across time. All right, let's recap 18.5. The first learning objective asks us to compare the various kinds of artificial immunity. So we had artificial active immunity produced by vaccination. This is when the immune system actively generates antibodies and memory cells. This gives us long-lasting protection, such as through the flu vaccine or MMR vaccine. For artificial passive immunity, this is when we get an injection of preformed antibodies, antibodies that were made by someone else. This gives us immediate protection, but it's temporary because we don't have any memory cells. It can also be used for post-exposure prophylaxis, for example, in rabies cases. So this is useful when you need rapid immunity or when the patient is immunocompromised. So again, for active immunity, you make your own response, you have memory, but in passive immunity, you're receiving antibodies and there's no memory. For the second question, we kind of answered it earlier, but let's review. Differentiate between variolation and vaccination. So variolation was the early kind of ancient or historical method to prevent smallpox. They used infectious smallpox material from scabs or pustules and introduced that to other people. That could provide a controlled smallpox infection and provide immunity, but it was very risky because it could also cause full-blown smallpox. It could spread smallpox to others, and there were high rates of morbidity and mortality. Whereas vaccination, introduced by Edward Jenner, used that cowpox virus, and it was related to smallpox but much milder. So this allowed immunity without causing smallpox. It was much safer, and there was no risk of transmitting smallpox. So again, variolation used live smallpox, whereas vaccination used non-smallpox virus. We used cowpox, making it dramatically safer. For the third learning objective, this is really referring to that table from the book, so let's look at that table again together. So that third learning objective asked us to describe different types of vaccines and explain their respective advantages and disadvantages. So I've blanked out those columns. See if you can go through each, provide a description and a few pros and cons for each of the vaccine types. And here's the complete table again, so you can compare your answers here. All right, and that takes us to the end of Chapter 18 on adaptive specific host defenses. Thank you for watching.